Hello class. Today we're going to be exploring the third line of defense. The third line of defense is the adaptive or specific response. This is specific to a single antigen. Our other two defense lines that we looked at, the first and the second, were non-specific. Right? They only looked at general defense mechanisms our body has. You're going to learn that the adaptive or specific immune response is very good. It's very strong. It's a very powerful system, but it is much slower. That's why we need this concerted effort between our first, second, and third line of defense. So, what does our third line of defense include? Well, it includes humoral immunity, which is effectively your antibody-mediated immunity. That's where your B lymphocytes will produce antibodies. And the other branch is the cell-mediated immunity. Now, cell-mediated immunity is mainly to deal with intracellular threats, whereas humoral immunity deals with everything. Cell-mediated is to deal with those intracellular threats like viruses or parasites like malaria, things like that, or cancer cells. Now, both B and T lymphocytes are produced in the bone marrow. We talked about this when we talked about blood. Just a reminder, all of your blood cells, all of your red blood cells, your white blood cells, as well as your platelets, they're all produced in your bone marrow. But they don't all mature there. We're going to learn about how T lymphocytes mature and that important step in developing immunity. So the third line of defense has three very important aspects. It is highly specific to a single antigen. It is systemic. It is completely system-wide. It acts in all parts of your body. And it has memory. This is why vaccinations work. Right? Your body has a memory to the things that it was infected with previously, and that allows it to respond much quicker upon reinfection. Now, the big downside is it takes longer to activate than non-specific responses. It takes much longer to activate. So let's talk about humoral immunity for a second here. Now, this antibody-mediated immunity involves B lymphocytes which do mature in your bone marrow. Now, these B lymphocytes are antibody-producing cells. They will turn into what's called a plasma cell, which is effectively just an antibody-producing factory. So once they become activated, they will just spit out antibodies day and night until they literally die of exhaustion. And antibodies, we learned about these in blood again. These are simply just Y-shaped molecules that have receptors, right? They have sites that can bind to specific antigens. That's all they are, right? They have these two receptors and they have a tail. So these antibodies are an essential part of your immune response. They mediate several different things, which we'll talk about in a bit, but they are absolutely essential in protecting you against infection. Next up is cell-mediated. This involves, instead of antibodies, it involves living cells as protective factors. And these living cells are T lymphocytes. Now, what is the role? Well, the role of these T lymphocytes is to recognize infected human cells and cancer cells. These T lymphocytes will attack these infected cells, quickly kill them, and continue to search for more cells. So these T lymphocytes are highly effective and specialized, the cytotoxic T lymphocytes that is, at killing infected and cancerous cells. Now, they mature instead in the thymus gland. And our thymus gland is like a military testing facility with the harshest of conditions, right? This thymus gland will absolutely ensure that we are not going to produce immune cells that can attack your own body. So effectively what your thymus gland does is it takes all of those T lymphocytes that were produced in your bone marrow and it says, okay, here is a bunch of different possible self-antigens. Do you react with them? And if there's any chance they do, guess what happens to that T lymphocyte? Yeah, it gets killed, right? It gets killed. So the reality is only about 2% of T lymphocytes graduate and don't get killed and go on to serve as immune cells within your body. So your thymus gland every single day is preventing a T lymphocyte from entering your body that would attack yourself. Don't get me wrong, it's not perfect. I know a lot of my friends have different autoimmune disorders, and autoimmune, you are attacking yourself, right? So our system is not perfect, but your thymus is very rigorous in weeding out those cells that could attack your own body. Alright, so T lymphocytes, we have two major kinds here. The first is your helper T cell, and these are one of the most important cells for your immune response. Without T helper cells, you cannot have a humoral or cell-mediated response at all, right? They activate both systems. So that thymus gland testing those T helper cells, that ensures that you do not have a humoral or cell-mediated response against your own cells. Now, the cytotoxic T cells, aka killer T cells, these are highly specific, they kill abnormal cells. So they kill cancer cells, they kill cells that are showing signs of infection, and they are very effective in doing so. Alright, helper T cells. They activate B cells and cytotoxic T cells, aka they activate the humoral and cell-mediated immune response, but they themselves must be activated first. Okay, so how does that happen? How do our B cells become activated? Well, our T cells, how do our helper T cells become activated? Well, our helper T cells become activated by encountering that antigen. When I say encountering that antigen, each helper T cell will only have receptors to one antigen. That's it. So they have these receptors on their cell surface that are specific to a single antigen, and once they encounter that antigen, they will become activated. Now, the most common way they encounter that antigen is through what's called dendritic cells. These dendritic cells will eat things like bacteria or viral particles or whatever else, and it chops them up and it presents pieces of them on their cell's surface. So these little pieces here, these antigens, helper T cells will come along and will say, no, I don't fit to that one. Oh, no, I don't fit to that. Oh, look at that. I do bind to that. If it binds to that antigen, well, guess what? Now that helper T cell is activated, and it will go now and it will look for B cells that have that receptor as well, and it will look for cytotoxic T cells that also have that receptor. So, the helper T cell needs to be activated before it will go on to activate your immune responses. Okay, so what about cytotoxic T cells? you're going to learn that there's a lot of checks and balances in this system because, again, you don't want to attack yourself. So how do they become activated? Well, all nucleated cells present antigens that represent what's going on inside the cell. So literally the majority of cells in your body, they're constantly taking things from inside of them and presenting them on the surface of the cell. Right? They're saying, look what's inside of me, look what's inside of me. And maybe what they're presenting is viral proteins, right? Maybe they're infected with a virus. So now they're presenting viral proteins, for example. And if a cytotoxic T cell comes along, it can bind to this. Remember, cytotoxic T cells, just like helper T cells, they have one receptor, one type of receptor on them. They can only bind to one specific antigen. So let's say it binds to that specific antigen. Well, now it can kill that cell. Keep in mind, these cytotoxic T cells will only be active if they have been activated by a helper T cell. So that's why there's multiple checks and balances here, right? The helper T cell comes along, it activates your cytotoxic T cell. That cytotoxic T cell then needs to bind to a specific antigen, and then it can kill that cell. So how does it kill the cell then, you ask? Well, viciously. Very viciously. It has these specialized proteins called perforins, and perforate to poke holes. That's literally what it does. These perforins will poke holes within the cell membrane. The cell membrane's contents, obviously the cytoplasm stuff, starts to leak out. And they also secrete something called granzymes, which tells the cell to kill itself. So it secretes things that pokes holes in it, and then it tells the cell to kill itself. You know, it lyses the lysosomes, which will effectively digest the cell from the inside out. So this is essential to killing infected cells in your body. Things like viral infections, there's parasites that can infect inside your cells, or certain bacteria that can, and also it fights against cancer as well. So, killer T cells kill virus-infected cells, cancer cells, and can kill bacterium-infected cells. And there's some other intracellular parasites and stuff like that it fights as well. At least once a day, your immune system destroys a cell that would have become cancer if it lived. Alright, activation of B cells and production of plasma and memory cells. So, this may be a little too hard for you to see. It is fairly small here. But follow along in your notes. So, just like T cells, your B cells must also be activated first, right? That activated helper T cell will come along, and you try to find a B cell that has the exact same receptor. It finds a B cell with the same receptor, and then it activates that B cell. So then that B cell that is now active, and again, it doesn't necessarily have to happen T cell then B cell. You could have it the other way around, like shown here, where the B cell finds a antigen first and then gets activated by a helper T cell. It doesn't have to be helper T cell activates and then B cell finds something. So here it shows the B cell finds an antigen, and it's like perfect. Now it's hunting for a helper T cell. The helper T cell comes, activates that B cell. Now the B cell is going to split into plasma cells and going to split into memory cells. Memory cells are longer lived and they hang out waiting for reinfection. Plasma cells are the ones that produce antibodies. And again, these are antibody production factories. They literally produce them until they die. So these antibodies have many functions, one of which is it makes them look really tasty to phagocytes. It is like a marker, kind of like a magnet, like drawing in those phagocytes for those phagocytes to eat them up. And lastly, those memory cells are essential for having a stronger and faster immune response if you are infected with the same antigen again. So let's quickly summarize the third line of defense. The humoral system is used to fight all threats, intracellular and extracellular, and it involves mainly the production of antibodies. That is the whole goal. We are going to be producing antibodies, and in the blood unit, I did call them immunoglobulins. That's the more sciencey name for them. Now, antibodies serve several functions. They attract phagocytes, which is a big one. It signals those things for phagocytosis. They activate something called the complement system. Our complement system is another way in which we can kill things like bacteria. It pokes holes in bacteria, causing them to lyse, causing them to burst. I'm going to link below a great video by In a Nutshell, and they're going to be on my website as well. I recommend you watch all of their videos on the immune system. They are fantastic, but they do have one specifically on the complement system, and it is fascinating. The complement system is another fantastic defense mechanism we have. And on top of that, they do neutralize certain toxins. So let's say you are bitten by a deadly snake. Well, if you go to the hospital, hopefully they have antivenom. What the heck is antivenom? It's antibodies specific to that venom. And those antibodies can bind to that venom and effectively inactivate it, neutralize it from having its effect. So antibodies are fantastic for fighting infection. Cell-mediated, a quick review. We fight intracellular pathogens or abnormal cells with cell-mediated immune response. And they do this by killing them with cytotoxic T cells, right? Cytotoxic T cells will poke holes in cells and also tell them to kill themselves. Pretty dark. Pretty dark. I think I'm going to end it there for today. Next lesson, we will be talking about immunological memory, how our body responds to a first infection and a second re-exposure. And it's going to lead into why do we have this epidemic of antibiotic-resistant strains of bacteria that are arising today. I know coronavirus is scary, but something that keeps me up at night is if we don't figure out another solution to fighting bacteria, at this rate, we will likely be out of usable antibiotics by about 2050. Imagine going to the dentist for a simple procedure or getting a cut or a scrape, getting an infection and dying, right? We're going to be going back to a pre-antibiotic world, which petrifies me. So stay tuned for the next lesson and have a wonderful day.